Hello, and thank you for joining this DNA Genotech webinar, Moving Forward with COVID-19, Trends and Challenges in Molecular Testing. I'm Jeff Graham, Senior Product Manager for Viral Screening and Surveillance at DNA Genotech, and the session moderator. In this webinar, the speakers will provide examples of collection solutions and testing strategies being deployed for COVID-19 detection, and will provide insights into the future of testing and surveillance as we navigate SARS-CoV-2 variants and the deployment of vaccines. The first speaker will be Scott Rabuka, Senior Director, Molecular Products at DNA Genotech, who will introduce trends and challenges in COVID-19 testing and DNA Genotech's validated saliva collection solutions. The next speaker will be Dr. Rafal Iwasiow, VP of Innovation and Technology at DNA Genotech, who will review evidence to support saliva as a sample type for SARS-CoV-2 detection. The final speaker will be Richard Uhlig, founder and CEO of Quadrant Biosciences, who will provide real-world examples of how Quadrant Biosciences has implemented innovative testing strategies for COVID-19. Following the presentation, there will be time for questions to our panel of speakers. So please add your questions in the Q&A chat box at any time during the webinar. And with that, I will hand the session over to my colleague, Scott Rabuka.
Thank you, Jeff, for the introduction. Thank you all for attending this webinar today. DNA Genotech is a subsidiary of OraSure Technologies. I wanted to take a few moments to give an overview of OraSure and who we are as a company. Within OraSure, there are three main solution areas. The first is within the diagnostic business unit, serving the infectious disease as well as the substance abuse testing markets. The next is related to sample collection and stabilization technologies for molecular applications. And finally, we have a molecular services business unit that focuses on data analytics, artificial intelligence, as well as a multi-omic view to health and wellness. Within OraSure, there are currently three main COVID-19 product areas of focus that are ongoing, two of which are within the diagnostic business unit, and those are the development of a COVID-19 rapid antigen self-test, as well as a COVID-19 oral fluid antibody ELISA test. The focus of today's talk, though, will be related to the third program within the sampling solution division, looking at sample collection devices for COVID-19 molecular testing applications. So with that, we'll move to the portion where we'll talk about sampling solutions for molecular testing of the COVID-19 virus. But first, I wanted to spend a few moments talking about trends and challenges that we're seeing. So from our purview, we provide solutions for collecting samples to molecular labs across the world. We're seeing some very stark trends related to moving from symptomatic to asymptomatic testing, looking at widespread programs for back to school, back to work, travel related and event-based testing. Testing labs are starting to confirm that their tests can capture the various variants of SARS-CoV-2 virus that are appearing. As well, we're seeing a broadening of the panel beyond just the SARS-CoV-2 virus to other flu viruses and respiratory viruses. And the big question for us is, as viral screening and surveillance progresses, what will be the new normal? You know, in 2 years from now, 5 years from now, when we're past the pandemic and there's widespread vaccination, will there still be testing ongoing? When you get a flu in a couple years from now, will you want to get tested so you know transmissibility of the virus that you may have, you know, what you can do, what measures you can take to keep others around you protected and healthy? So some of the challenges to getting here are really around providing safe sampling solutions, getting widespread adoption, and also population adherence to COVID screening programs. So what we're focusing on is ensuring that solutions are in place that can in fact be used by anyone, anywhere, anytime, and in any environment as well, so that There's non-invasive solutions that are safe for transport, safe for handling, and so on, which we'll be focusing on for the rest of this talk. So within the DNA Genotech portfolio of products, here are the product areas that are specific to COVID testing. So there's two main form factors. One is where the patient will provide a saliva sample. That's within the OmniGene Oral and the Origin DX products. And the other is related to the OraCollect RNA product. The first product requires 1 or 2 mils of spit, and the second product uses the sponge connected to the tube to collect the sample from the mouth. Both product lines have similar value propositions in that they're easy to use, they're safe for home collection. Rafal will be talking about the inactivation of the virus, and they also both have a non-toxic chemistry, making handling safe and harmless, whether you're the one collecting the sample or handling the sample within the lab. Both products stabilize the virus at room temperature and can be used for ambient transportation from the collection point to the lab. Both products have a tube where the sample is collected in that is compatible with high throughput environments within the lab. And they can also be used with PCR and sequencing applications. From a regulatory status and intended use, the products have been approved by the FDA under the emergency use authorization process, and they can be used by individuals to collect, stabilize, and transport saliva samples or molecular testing of the SARS-CoV-2 virus. Health Canada has authorized them for at-home self-collection for COVID testing. And both products are also CE-IVD Class 1 registered products within Europe. So I want to take a moment to talk about and show how the samples are collected with both devices. So OmniGene and Origin, as I said, are both a saliva-based collection kit. The individual or patient will spit 1 ml of saliva into the device up to the fill line, and then the cap, when it's pressed down, there's a film protecting the chemistry. It will pierce the film and the chemistry will mix with the saliva sample. In step 3, you remove the funnel and lid assembly, and in step 4, you place the cap on and then you shake it to prepare the sample for transport and then ultimately arriving at the lab ready to be put into an extraction workflow. OraCollect RNA uses a sponge to collect the sample. You place a sponge in the gum or cheek pouch of the mouth on one side and move it back and forth 10 times, and then you move it to the other side and do the same. Then you invert the tube cap and the sponge is placed inside the tube and you mix it 15 times back and forth. And again, the tube is ready, sample is stabilized, and can be shipped to the lab for processing. So what we're showing here is a typical workflow for a COVID-19 molecular test in which the sample is collected at the point of collection. It could be at home or at a collection center, and then it's shipped to the lab for extraction, where following extraction, an assay will be run for detecting the SARS-CoV-2 virus. And then from there, you can go to public health action, so reporting positives and negatives, for instance. The technologies listed below extraction and the assay were all included in one or more of the EUAs that were approved using our collection devices. There are now more than 180 academic and commercial customers using our devices within their workflow for SARS-CoV-2 testing. This slide shows a list of all 8 test providers that have received an EUA for their COVID-19 testing using our collection kits at the front end. So the first column shows the lab, second column shows which DNA genotek device was used. The third column and fourth column show the extraction and testing platforms, and those are referenced on the previous slide. And then the last column shows the use that was approved. So in some cases it was supervised collection, others were unsupervised collections for clinical or at-home use. Rafal will get into more details around the studies that went into many of these EUAs and that have gone into validating the devices for SARS-CoV-2 testing. So at this point, I'd like to pass it over to Rafal, and again, please bring forward your questions towards the end and we'll do our best to answer them. Thank you.
Thank you, Scott. I'm happy to share with everyone now some of the data supporting the use of our devices in COVID-19 testing. Here we have two examples of clinical data from two different EUAs. On the left, you have Quadrant Biosciences EUA for the Clarified COVID-19 test kit. Below you see the clinical data comparing saliva to NP, nasopharyngeal swabs, with 100% concordance both in positive and negative samples. On the right, you have data from the DNAGenome OME 505 EUA. Here, using the CRL rapid response test, which has also received an EUA for COVID-19 testing, we show a 100% concordance between saliva collected samples and nasopharyngeal samples. We have also done significant amount of work internally. On the right, you see stability data from samples kept at room temperature for multiple days or simulated transport. Simulated transport means samples that have been cycled between freezing conditions and elevated conditions. In fact, we recommend that our samples collected in OME 505 or ORE 100 be heated at 50 degrees when they arrive in the lab. This activates our chemistry and makes sure that the samples are homogeneous. Homogeneous samples improve reproducibility of sampling. Due to the robustness of our chemistry, some customers have elected to heat at even higher temperatures, 56 degrees Celsius. This helps to decontaminate exterior of the collected devices without compromising the quality of the RNA. We have also done significant work on inactivation. The chemistries in our devices, specifically the OmniGene Oral and the OraCollect RNA devices, are capable to inactivate SARS-CoV-2. Due to some limitations of the assay, we claim greater than 99% inactivation. On the right, you see a positive black bar. This represents virus spiked into Vero6 cells, and the negative control, the hashed bars. The gray bars represent virus, which was spiked into our chemistry, and then into Vero6 cells. You can see that the level is identical to that of the background negative control. We have also been supporting EUAs by leveraging years of experience and robustness from our FDA 510K cleared Origin DX devices, specifically the OGD 500 and the OGD 610 device. These devices share the physical features of the OME 505 and have the same instructions for use for collecting saliva. On the right, you see one example of such a robust study. In this case, we're looking at a diverse demographic of individuals and using volume of saliva collected as an endpoint. In this particular study, 99.7% of study participants correctly followed the instructions for use, and 98% of participants collected the appropriate volume of saliva. we can leverage the data I just presented on stability, inactivation, usability to support the use of OmniGene Oral, or GeneDx, or Collect RNA for use in COVID-19 workflows. In fact, these three devices have been included in multiple EUAs, and they use a permutation of extraction and assays, showing the versatility of these devices. In addition to the work that's already been submitted and authorized through EUAs, we have been doing additional work. Here is an example of a reagent we call PrepIt Q2A, Quick to Assay. This is a simple sample preparation solution that helps to bypass traditional extraction. This is purely a liquid-based processing, takes 30 minutes start to finish with as little as 5 minutes of hands-on time. It has been custom formulated to work with our ORE100 and OME505 devices. This process helps to remove the inhibitors present in our chemistries and in saliva, and allows then the sample to be used directly in PCR, NGS, and microarray. We're happy to work with customers on further evaluation of this chemistry in your particular assays. Another use case for saliva is asymptomatic testing, reaching a large number of the general population. Here's an example of a recent publication submitted to meta-archive. On the right, you see some data from that publication. They tested across asymptomatic positive and negative samples using 2 EUA assays and showed 100% concordance between those assays. Additionally, and interestingly, sampling across a large number of samples, approximately 9,000, in both an asymptomatic and symptomatic population, they show that the CT values between these two populations are very similar. Pulling of saliva samples for COVID-19 testing is another use case that can be enabled using our devices. Our devices, which provide long-term stability to the samples and give a homogeneous sample to work with, help to enable pulling. Here, I share with you 3 independent publications that support the use of saliva for pulling approaches. The FDA has a pulled sample testing and screening testing for COVID-19 guidelines that I encourage you to review on how to proceed with sample pulling. DNAGenome Tech is supporting its client by supporting the adoption and validation of saliva and testing labs, working closely with extraction and assay vendors to develop optimized protocols using DNAGene to collect devices, leveraging previous experience in collection and stabilization, generating technical data to support the use of DNAGene chemistry, and working closely with regulatory approvals required by test providers. Thank you.
I'd now like to invite Richard Uhlig, CEO and founder of Quadrant Biosciences, to introduce Quadrant and how they pivoted to address the COVID-19 pandemic. Over to you, Rich.
Great, thank you so much, Jeff. Hi, everybody. I hope everybody's safe and healthy today. I'm pleased to introduce Quadrant. Until COVID-19, Quadrant was an epigenetic diagnostics company with a specific gear toward neuroscience applications. And we were really trying to drill down on two different areas. One, a basic science question, which is what is the origin of some of the molecules that we see in saliva, master signaling molecules like microRNAs that are of interest to us, and we have very compelling evidence on that, that those molecules are in fact brain derived, the ones that we're interested in, and actually follow the same pathways as the rabies virus, which you may know replicates in the brain and travels via the 5 pairs of cranial nerves into the oral pharynx and into the saliva. The second question was really related to translation of that information. So, and we've spent the last several years building large scale data sets for some pretty large problems in society. One being, how do we detect children on the autism spectrum at the earliest ages? We've made tremendous progress on that, and we've happily used the DNA Genotech collection kits to help us with the collection of saliva samples from those children, and then using next-gen sequencing to determine the quantification of those molecules of interest, and then using AI and machine learning algorithms to help us differentiate case from control. Our work spans beyond autism spectrum disorder to concussion injuries, or MTBIs, Parkinson's, anorexia nervosa, as well as some nascent work in schizophrenia. So as we came into about this time last year, it was pretty clear that if we were going to best serve our communities, we needed to pivot into COVID-19 diagnostics. And we saw three areas that needed direct help. One is giving people confidence in their own health. In other words, developing an individual diagnostic, helping those individuals have confidence in the health of those around them. And that was development of a screening or surveillance type methodology. And we did develop a pooling technique relative to that. And the third is to have a better understanding of the dynamics around policymaking. And we saw the development of a wastewater surveillance program as being key to that element as well. We did, in fact, develop all three of those areas, and starting with wastewater. We do have a very active wastewater surveillance program covering about 500 different colleges, universities, municipalities, et cetera, across New York State, Northern Pennsylvania, and Vermont. That's a lab, an environmental lab that we set up in within SUNY Upstate Medical University in Syracuse to handle that product line. Secondarily, we developed a saliva-based individual assay that we then also iterated into a pooled testing tool. So the individual assay, it's as you saw on the previous screens, it's called the Clarify COVID-19 test kit. We are a manufacturer of that. We use that ourselves within our labs, and we have built two high throughput COVID-19 labs in upstate New York, but we also sell that to third parties. And that test kit, we did focus mostly on quality. Because we, as again, some of the research that was put up on the screen, the quantification of the SARS-CoV-2 virus in saliva, understanding what portion of the population your test will be effective with is pretty important. So I'm pleased to say that the Clarify COVID-19 test kit is the #1 most sensitive saliva test in the world. We detect as few as 600 copies of the virus per ml of material, and that's available on the FDA LOD website, and then I think we're tied for 6 globally, regardless of what type of specimen was collected. We then, with the focus on quality and understanding that reagents and testing materials are in short supply, and seeing critical issues relative to supply chains, we also then developed a pooling structure, so we can pool up to 12 people together into one PCR well. Unlike some of our competitors like LabCorp and Quest, who simply dilute the sample that goes into the extraction and amplification process, we actually take a first step and concentrate the RNAs before we go into the extraction and amplification processes, which allows us to get great sensitivity, even in a pooled basis. So, for example, we have a pool. If we have even one infected person in a pool of 12, our CT threshold for that pool will end up being very similar, and it's typically a little bit. It's within one cycle of what that individual sample would be on its own. So we've got great technology on that front. We've deployed this technology pretty broadly, mostly again across New York State, but we are now having clients nationwide. We've sold about 3.6 million clarified COVID-19 tests since September, when we received our FDA EUA, and then within our own labs, we've processed about 1.4 million tests as of today. So, and those labs are located in Syracuse, and we're just about to fire up the lab in Buffalo, New York. Each one of those labs will have capacity of about 25 to 30,000 people a day. Currently, our Syracuse lab is running anywhere from 15 to, I think our peak was about 28,000 people in a single day. So that's what we've brought to the table. We're a relatively small company, but as my colleagues like to say, we're small but fierce. So, thank you, Jeff.
Thank you, Rich. That was a great introduction. Much appreciated. So we've now arrived to the question and answer section of the webinar. So if you have any questions, please add them to the Q&A chat button, bottom middle of the screen. We did have a few questions come in before the webinar, and a few of them were related to various aspects of the state of the art in testing. And we had a question about antibody testing and assays and the most accurate assays available, as well as a question on detection instruments and in particular point of care testing. So I think that I'd like to start with a question more broad, but really on how we've seen the testing landscape evolve from early pandemic to now, and any insights into the future of COVID-19 testing that you might have as vaccines roll out, and different mix of testing may be important. So maybe I'll punt that one to you, Rich, to start, and then maybe Rafal and Scott could jump in.
Yeah, I think the landscape has changed pretty significantly. If you roll back to the summer, most labs they didn't really care about the quality of testing. They were just concerned about, can they get a platform where they can get sufficient reagents and be able to actually run tests for their particular clients. That's since changed. A lot of the supply chains are in reasonably good shape right now. I will tell you that there's we typically are stockpiling about 100 to 200,000 reagents, primers, probes, filters, whatever, and collection kits for about a 1 to 2 week period in front of what we're seeing. We have had to develop strategies very quickly, get them validated and through the FDA process to be able to deal with some of the supply chain issues. But I do see some pretty significant changes on the horizon as greater percentages of the population are being vaccinated, and we see changes in the testing itself as well. Right? There's been some some of the tests that use, for example, the N gene, the N1, N2 probe and primer set. Some of those are now less effective in detecting the virus as the virus continues to mutate. Just as a reference point, we use the probes and primers that were developed by the Pasteur Institute, and those have been really, and we continuously test those versus the new variants to be sure that our test maintains its effectiveness. We do see a migration toward lower, like higher throughput and lower cost testing, which again is toward pooled testing that can accommodate that, but then also some of the other elements that were talked about earlier, self-collection, at-home collection, pooled testing, as well as asymptomatic testing of populations, all of which we've either received authorization from the FDA or in flight relative to that. The two other areas that I'd like to point out are that we're going to be adding to our clinical labs are specific antibody testing, and we are working closely with a team at OraSure relative to that. And then the second area is in digital representation of health, and we as a company are working with IBM for their digital health passport. More and more of our clients, and particularly large-scale large-scale sporting venues, want want their population, whether it be fans or athletes, to be able to show on their smart device their relative COVID status, which is kind of a constellation of three things: their vaccination status, their antibody titer levels, as well as their status relative to the virus itself.
That's great. Thanks, Rich. Scott or Rafal, would you like to chime in?
Yeah, no, that was a good overview from Rich. I think beyond from a testing technology perspective, certainly we're seeing more groups look at sequencing and moving from a single COVID test to a respiratory panel, as well as ensuring, as Rich mentioned, that the variants of the SARS-CoV-2 virus are captured as well.
Yeah, I would also just add that we, because of the volume that we're doing, and we are averaging about 20,000 people a day, we do take a portion of those positives that we have, particularly if it's a high titer or if it looks like it's part of a cluster, and we do sequencing analysis on that to understand essentially what portion of our positive population is the original Wuhan strain versus any of the other variants that we're seeing come into our testing population.
I think I would just add that I think we've seen a pretty significant transition from where samples are being collected and where testing is happening. We've got a lot more options now. We're initially it was nasopharyngeal swabs, and now we've got saliva options that Rich has outlined, as well as antigen tests at the rapid tests at the point of need. So I think there's a lot more options available now. I think to dovetail on that last question, and I think Rich, you alluded to a few few of these areas, what are some novel ways that labs and testing programs are reaching and testing more people? Given that lab capacity and trained healthcare workers and personal protective equipment is limited, what are some of the ways of sort of creating more efficiency and reaching more people?
Yeah, that's a great question. So I think the work that the DNA Genotech team actually did to allow for self-collection was certainly huge for our program. So, for example, in a college campus, we can now have a distributed labor model where we've got staff on campus staff, not Quadrant staff, but on campus staff actually facilitating the collection process and then helping ship those collection tubes to us at Quadrant for processing. So we didn't have to build that labor force around that. So the self-collection is a huge thing to facilitate multiple different venues. And then the second thing is really the pooling process. So for example, if anyone has a lab and you've got say PCR wells enough for 1,000 patients in a particular day, by migrating to pooling, you've immediately turned that into 12,000 patients a day. So you reduced your overall footprint in the supply chain. So that's really been one of the big advancements to get larger, broad scale testing done out there. And at an affordable price. So.
Great, thanks, Rich. We have a few questions coming in. I'm going to direct this one to Rafal. Does PrEP Q2A allow for testing a sample without extraction?
Yeah, that's exactly what the intent of that product is. It really depends on the assay you're using. So how sensitive your assay will be to any carryover of potential inhibitors. The chemistry does a great job removing the known inhibitors that we have in our chemistry that give the stability to the sample. And it does a good job also removing the environmental inhibitors, but sensitivity of the assay is key here. So I would encourage, direct testing. I can tell you that we've tried microarray sequencing and several different RT-PCRs, and we can certainly get it to work. Works well, but performance will really depend on your specific assay, and we're happy to work with customers on that.
Great, thank you, Rafal. I think a second question for you that just came in. How much does food and liquid intake and tobacco smoke affect SARS-CoV-2 in the sample? And what are recommendations for collection and avoiding any impacts?
Yeah, that's actually a great question. I'm not sure how robustly the impact on SARS-CoV-2 has been tested in these situations, but what I can tell you, we've done a lot of testing with these devices as far as it goes for food, drinking, tobacco, anything, many other compounds. We call these interfering substances, potential interfering substances. We've done a lot around the total nucleic acid aspect and shown that we can effectively take care of them, remove them through extraction, and the chemistry takes care of all those things. So we have not seen any carryover of this as impacting RT-PCR or PCR reactions.
I can tell you that as part of our FDA EUA, we did a lot of work relative to interfering substances as part of that requirement. We were successful in that. However, we do ask people that have had food, drink, tobacco, whatever, 30 minutes prior to testing, that they wait an hour before actually testing to help us with continuity of test samples.
Yeah, that's a great point, Rich. Our standard protocol is at least 30 minutes wait, and that's because you just can't predict everything that a user might do.
Great, thanks, Rafal. I've got a question about, it ties into the current and the future testing needs around safe return to workplaces, schools, and travel. And I guess start with you, Rich, and then Scott and Rafal on, I guess approaches being used, and you mentioned pool testing, but what sort of, evidence supports testing and asymptomatic and asymptomatic testing? And maybe we'll start with that.
Well, for asymptomatic testing, we all of our college, university, K through 12 programs are really asymptomatic testing that we're doing, right? If anyone has symptoms, we're running and I'm sorry, that's for pooling. For anybody that is symptomatic, we're running just a straight individual test for that person because it doesn't, you would lose the economies of doing pooling if you have known symptomatic individuals in that. That's been a great program. It's been so cost efficient that the state of New York, State University of New York system, which is the United States' largest higher education system, 64 campuses, 450,000 students. In normal times, about 200,000 students and faculty and staff and residents currently are doing testing every week on that population. The net result of that has been that unlike the common narrative that college students are the vector of transmission, what we've seen is in fact the opposite. So, and again, I think that's somewhat a function of our test being so sensitive as well. But the positivity rate on the SUNY campuses in almost every instance is significantly lower than that of the community around them. So it's been great evidence that this type of testing program works pretty well.
Yeah, I would just add that we're seeing similar testing programs in other states and other venues as well. I think I think early on there's probably a stigma with getting a COVID test, and it's becoming in some environments a staple of high functioning society, if you will, or your workplace or school campus or whatever. Knowing where the virus is and how it may be spreading is really important to containing it, and as programs like the one at SUNY catch on, there's a better chance that the nature of spreading and understanding where it's coming from and how it can be contained is going to be much more actionable, I'd say.
Great, thanks, Scott. I've got another participant question that I'll direct to you, Scott. And the question was, what percentage of labs process tubes manually versus using automation?
Yeah, so I think we really only saw manual processing from labs during early feasibility and validation work. Any lab that's gone to scale with our device, we believe, are using automation, and I think that speaks to the fact that the tubes are standard labware. They fit within a workflow fairly seamlessly. The major robotic manufacturers have racks and instrumentation that are compatible with the lab, so it's not hard to automate the processing once they get to the labs. And if there's a specific instrument that the person asking the question is interested in, we can certainly address that and work with the manufacturer to show compatibility as well.
Excellent, thank you. And a reminder, if there are any questions, please add them to the Q&A chat box at the bottom middle of your screen. Since the session was really on trends and challenges, I want to get to some of the trends and a little bit the future around, the importance of detection of variants and, going forward, environmental screening. So I'm wondering if the panel could comment on how labs and public health agencies are responding to the new variants that are being detected. And, where do we see surveillance going in the future to prevent a pandemic like this from happening again?
Yeah, in my in the slides that I shared earlier on, I pitched the idea of what will it look like in the future. So let's say we get past the pandemic, widespread vaccinations in place. Rich, I'd love to get your take on if you have a flu in 2 years, because you're able to know now, will you go get tested to figure out what strain of flu it is and what it means? And
yeah, I do think that we're going to be, because of the global nature of the coronavirus. I do think that this is going to be something that we are working through and dealing with for years to come. It's none of the vaccines seem to be universally effective. So there's and as different variants emerge, like for example, I believe that the South African variant effectively evades the Moderna vaccine. I think I have that correct. But I think that there's going to be ongoing surveillance relative to the variants, and just like with the flu in years prior, there will be a vaccine that our public health experts best guess as to what over the coming year will best protect the population relative to that particular variant. But I think that's probably the future that we're looking at. So yeah, there's going to be ongoing testing. Some of the work that we're doing in wastewater is particularly interesting because we can start to see we can predict hospitalizations by about 5 to 7 days by seeing various levels of the virus in wastewater, and we're working on a program right now to better understand what the proportion of different variants of the virus are in that wastewater. So, and that's hopefully we'll hear today whether we got a large-scale HHS contract relative to that. But I think that's part of the future to be able to understand how different variants transition across a population or emanate out from epicenters.
Great answer, Rich. Thanks. I think with that, I'm not seeing any other questions. Unless any of the panelists have any closing remarks or items they didn't get to, I'm going to, I'm going to finish by thanking Scott, Rafal, and Rich for presenting and answering all the questions today. If we didn't get to your question, we'll make every effort to reach out to you following the session. And contact details are here, so also feel free to reach out to any of the panelists. I'm sure they'd be happy to speak with you. And there's a link at the bottom there as well if you're interested in validating your test with any of our kits. They're available for free at the link there. And with that, I will close the session. Thank you.